Thank you, everybody, for coming for the second day of lectures from our guests, for Professor Lise and Peter Nigrovic from Howard University. The first lecture will be on megakaryocytes. It's a very interesting topic and the role of these cells in immune system. So we invited Professor Peter Nigrovic to give this interesting, I'm sure it will be very amazing lecture on this. Thank you, Peter.
Thank you very much, Professor Martini. So I'm very delighted to be here to speak with you. I'm very sorry we're starting late. We had a little bit of technical problems given the size of the file, but thank you for putting up with the delay. So today, I'm a rheumatologist. So I do internal medicine and pediatrics, and I do rheumatology, so joint and immune diseases. And so typically rheumatologists don't think about megakaryocytes very much. But I also run a laboratory, and in the laboratory, we can pursue things that we find interesting, but that are not directly clinically related. So what I'm going to tell you all today, nothing's going to be on the test, which is to say there's nothing here right now that you're going to have to know for your clinical practice. But I think, here we just toured your beautiful new research building with mouse space and translational space, and you have so much potential here to sort of pursue issues of curiosity, and these lead to interesting directions. And more, it's that kind of story that I want to tell you. I want to tell you sort of an interesting story that's sort of a basic science story. So I don't have any disclosures related to this talk. So megakaryocytes are cells that make platelets. We all know that they live in bone marrow, and they start as a hematopoietic stem cell. They then undergo a process called endomitosis, where they increase their nuclear material, presumably in order to give them a lot of substrate to make a lot of protein and membrane, which they have to do to become very large and to make platelets. Then they stretch out their membrane into what are called proplatelets, and these proplatelets are released as dumbbell-shaped structures that then are sheared in the circulation into platelets. And these are the source of our platelets in circulation. And so this process is really a very beautiful process. So here's a video of a megakaryocyte turning itself into platelets. And you can see I'll call attention to what a membrane-intensive process this is, because this is a big round cell having to turn itself into lots of little round cells. And that's going to be important because in order to do that, megakaryocytes have to have a very interesting membrane dynamic where they have a membrane reservoir inside their cells so they can immediately make all this membrane or provide it to the platelets. So when we think about our histology class, in medical school, and you look at the bone marrow, and you see different kinds of cells in the bone marrow, and one of them is this big round cell, which is the megakaryocyte. They're relatively uncommon, 1% or less of the cells in the bone marrow. And we think of them as just sitting there in the bone marrow, but that's not quite true. So megakaryocytes can actually leave the bone marrow. And this was discovered initially when people were looking for cancer cells in circulation. When they found big round cells, they found that they were actually sometimes megakaryocytes. So megakaryocytes can not only happen in the bone marrow and in spleen through the process of extramedullary hematopoiesis, but they can also migrate, and they migrate to lung because when they leave the bone marrow into the venous circulation, they get stuck in lung. And that happens pretty frequently, something like 250,000 cells an hour in humans. And in fact, some proportion of platelets, we don't know quite how many, are made in lung, as the megakaryocyte immobilizes itself in the small capillary membranes and then shears off its platelets in the circulation. And here's an example. This is not work from our lab. This is from John Looney's lab. This is looking at a live mouse. So this is called in vivo 2-photon microscopy. The red material is blood vessels, and the green is platelets. And you can see here that there are lots of little platelets, but there are also these big events, which are megakaryocytes flying into lung and then shattering into platelets. And this happens with some regularity. So these are videos from this paper published in Nature a few years ago. So we got interested in megakaryocytes for another reason. So my lab does animal models of arthritis, among other things. And we use a particular model in which you can model a process related to rheumatoid arthritis, which is, at least in some sense, is an IgG-mediated arthritis. So in this model called the KB/N model, you take serum from one mouse that has arthritis and you put it into another mouse, and that mouse develops an immune complex arthritis. And we were looking as to what lineages, what cell types are important in that process. And in this funny mouse that is lacking, has a mutant receptor for the stem cell factor receptor, it's resistant to arthritis, but that resistance can be overcome by engrafting different lineages, mast cells, for example. And we found that if we engrafted megakaryocytes, but not platelets, we can actually restore arthritis susceptibility to that mouse. So if we put in wild-type megakaryocytes, we would see that the arthritis came back. And if we put in interleukin-1 knockout megakaryocytes, but not platelets, we would see that the arthritis did not come back. So the megakaryocyte in this model was providing a systemic interleukin-1 signal that was enabling arthritis to happen. And it had previously been shown that megakaryocytes make microparticles. These are small blebs off the membrane, which it releases in abundance into circulation. And we discovered that these microparticles had interleukin-1, both interleukin-1 alpha and interleukin-1 beta. These are very potent pro-inflammatory cytokines. And if we took megakaryocytes and put their microparticles on cells that were informative as to whether they were seeing interleukin-1, you could see in the blue bars where the receptor cells were lacking the interleukin-1 receptor, they wouldn't respond with that pro-inflammatory signal. But if they had the interleukin-1 receptor, the clear bars, you would see that they developed an interleukin-1, they developed an inflammatory response. And so this led us to think about megakaryocytes differently. So we've long thought about platelets as pro-inflammatory factors. We know that platelets have mediators. They help neutrophils to migrate. They cause neutrophils to degranulate. They have all sorts of anti-infectious properties. But we didn't think of megakaryocytes that way. And so we started to think differently about megakaryocytes as immune cells. And we summarized that in this review, noting here in this model megakaryocytes that megakaryocytes express cytokine receptors. They express different things like CD40 ligand, which are immune receptors, FC receptors, toll-like receptors, etc. And they can make not only platelets, which we knew were pro-inflammatory, but microparticles, which we showed in our paper were pro-inflammatory. They can cross-present antigen on MHC class I, so they can be antigen-presenting cells, and they also can make cytokines. So this is sort of interesting idea of megakaryocytes themselves as immunologic cells. And that set us off on the question, just a curiosity-based question, what else do megakaryocytes do in immunity? And that's what I want to tell you about for the rest of this 45 minutes or so, 40 or 45 minutes. And what I want to tell you about is something that's rather obscure. It's called emperipolesis. And this is a made-up word which comes from em, peri, and polemi, inside, around, wander about. And it was an observation some years ago where neutrophils, if you take a wet preparation of bone marrow and you happen to see a megakaryocyte, sometimes you see what looks like a neutrophil swimming around inside the megakaryocyte. And so this is a confocal image. So this is that very thin slice of bone marrow from a patient of mine who has the disease we talked about on Monday, which was systemic juvenile arthritis. So this is a very inflamed patient. And here in the bone marrow, you see green, which is the megakaryocytes. You see how it's a big cell. You see red, which is neutrophil membrane. So this is actually CD66B, which is a marker for neutrophils. And you see blue, which is nuclei. And what's remarkable in this section is you see the megakaryocyte itself with its own nuclei. These are these bluish blobs here. This, as you know, the megakaryocyte is polyploid. Oops, that's not right. Is polyploid. So I can't quite tell. So, but anyway, you see it up there. See that, and then you see inside it, in this confocal image that is really truly inside, not on top, not on the bottom, but inside are neutrophils. So what is this process of emperipolesis? So it had actually been described, as I mentioned, years ago in this paper in 1970, but it had essentially never been studied as to what it meant. You see it more in some diseases than in other diseases, but what it was doing was not clear. And what's interesting is it's evolutionarily conserved in all mammals. So there's an interesting thing about mammals and megakaryocytes. So if you are not a mammal, you don't have megakaryocytes. But if you are a mammal, you have megakaryocytes. It's a very close one-to-one relationship. And what's interesting is that emperipolesis is present in every mammalian species. So over hundreds of millions, over more than several million years of divergent evolution, this is a cell-cell interaction which in some region has been preserved, suggesting it has some kind of important evolutionary function. And so one of our goals is to try to understand what that function is. So we started with some initial observations in mice. So this is a mouse bone marrow. You see these big megakaryocytes, and you see that some of the megakaryocytes have neutrophils within them. Those are the red arrows, and some don't. If you put a mouse through an inflammatory arthritis, the arthritis I was talking about, KB arthritis, the megakaryocyte emperipolesis rate in a 5-micron section goes from about 3% or so to about 6%. So this is an inflammatorily induced reaction. And in fact, the slide you see on the left, where one megakaryocyte has 2 neutrophils in it, that's almost only in an inflamed mouse. You can see 1, but almost never 2 or 3 or 4. Like you can in an inflamed mouse. And that's true across models. So the model on the right is something called cecal ligation and puncture. This is the model of peritonitis, where you do surgery on the animal, you poke a hole in the intestine, and bacteria leak out into the peritoneum. It's a model of peritoneal sepsis. And those mice also have this increase in peritonitis, as does every other inflammatory model that we've tried. So it's an inflammatory reaction very commonly induced through inflammation. So how common is it really? So if you're looking at a slide, the paraffin slide that you've stained, that slide is 5 microns thick, so 5 micrometers. But a megakaryocyte can be 100 micrometers tall. So you don't really know from one section how often this occurs. So what we did was stain bone marrow in 3 dimensions, and that's what's shown here. So here is a bone marrow section. Now the megakaryocyte, you can see, is sort of a round cell. The white areas are the sinusoids. So these are the blood vessels in the bone marrow. And you can see the megakaryocytes pushed up against the white sinusoid. That's where it releases its platelets. And so inside the megakaryocyte, as we take the green color off, you see this neutrophil nestled. And using this kind of 3-dimensional imaging, you can see that at baseline, the real proportion is about 9% in the healthy mouse, and it about doubles, or a little bit more than doubles, in an inflamed animal. So this is not a rare thing. This is a relatively common relationship, especially in inflammation. So in order to understand what was going on, we decided to try to make an in vitro model, because it's hard to study things in vivo. So we took a bone marrow from a normal mouse, and we took megakaryocytes that we cultured from a normal mouse, and we put them together. And what you see is on the right, and that is a megakaryocyte. That's the big round green shape. And inside the megakaryocyte are many neutrophils. And sometimes you would see up to 30, 40, 50 neutrophils within a megakaryocyte. So you can reproduce this phenomenon very readily. And what I'll call your attention to is that there are 2 compartments that these cells seem to be in. So the first, here is the membrane of the megakaryocyte in green. I don't think I can't point, but I'll just point with you. You can imagine that I can point. So you see the green lining, that's CD41, that's GP2B, that's a membrane-specific marker of megakaryocytes. And so some of the neutrophils are in vesicles that seem to be surrounded by CD41, and others don't seem to have CD41 around them. So what is that? So to understand this better, we then did electron microscopy. So you do here, let the reaction go in vitro, stop it at some point, and then do sections and by EM. And EM allows you to see membrane, which other systems don't. So here we see what we'd expect. So here's a megakaryocyte, big megakaryocyte, and a vesicle. We call this vesicle an imperisome for reasons just because it's a little bit just something interesting I'll tell you about in a second. And inside the imperisome is a neutrophil. So this looks like a cell being phagocytosed. That sort of fits. Then we see images like this. So here's a neutrophil inside an imperisome, and the membranes have started to collapse together. So here on the right, we see two sort of train tract membranes. So they're each a bilipid membrane. So the one on the top is a megakaryocyte membrane, and the one on the bottom is a neutrophil membrane. So these are membranes smashed close together. And the arrows point to areas where they seem to get fuzzy, and we don't know if that's an artifact or if that's something real. And then what's really striking, and what was very surprising to us that we had to see many times before we believe it, is one membrane between the cells. This turns out to be the neutrophil membrane, not the megakaryocyte membrane. So this is, the neutrophil has penetrated through this imperisome and is now in the cytoplasm of a megakaryocyte. So this is, to our knowledge, the only example of a phenomenon like this, where a host cell penetrates into the cytoplasm of another host cell. We know pathogens do this, right? A lot of intracytoplasmic pathogens, but host cells don't do this kind of interaction. Even when they pass through each other, they stay topologically outside. They stay in a membrane-bound space, but here it's inside the cell, the cytoplasm itself. And we think that's what's going on here. So remember this image that I showed you before. Some are inside a CD41 bound vesicle, and some don't have CD41 around them. And we think that's because they don't have a membrane around them at all. They are now in the cytoplasm of these cells. So what is the fate of a neutrophil that does this? And so here, the red is the megakaryocyte, and the green is the neutrophil. And you can see the neutrophil pushing its way, what looks like pushing its way inside. It almost looks like it's looking normal migration. And so here, over the course of this 25 minute video, you see the neutrophil enter and then leave. So this is a transient reaction. And in fact, we don't hardly ever see a dead neutrophil inside a megakaryocyte. This is not a place neutrophils go to die. This is a place neutrophils go to pass through. So the question is, why would they do this? And the first clue we had was from that video that I just showed you. And here are the stills at 0, 4, 8, 12, 16, 20, 24, and 28 minutes. And if you follow, and the trick here is that this stain on these cells is a membrane stain. So what we're following when we see green is neutrophil membrane. And if you look as the neutrophil passes into the megakaryocyte, what it does is it leaves some of this green membrane behind. That is, there's a transfer process going on here from one cell to another. And in fact, we see that also by electron microscopy. So you remember that megakaryocyte that was turning into lots of little platelets, that beautiful video is from Joe Italiano. And that video that showed you how membrane intensive this process is. And the reason the megakaryocyte can do that is because it has a reservoir of membrane. Sort of keeps a kind of a storehouse called the demarcation membrane system in the cell. And the neutrophil turns out to migrate over to the demarcation membrane system and start to make contact. So here in the bottom image, you can see that there's the neutrophil is actually joined membranes with the demarcation membrane system and is feeding its own membrane into that system. And we think this is why the neutrophil has to be cytoplasmic. So if it were in a vesicle, of course, it couldn't make membrane-membrane contact because it would be sealed off. But in the cytoplasm, it can do that. And so now, is that real? Is this just like an EM artifact? You can't really tell by EM to make sure. So what we did was we biotinylated the surface of the neutrophil and then fed it to megakaryocytes. So biotin is something that can easily it can stick on the surface of the cell, and you can come back with a chemical called streptavidin, which has a label on it, and then you can see, is the biotin there? And so what you see then is if there's transfer, you should see that if the proteins, these little brown dots, appear on the surface of the cell, we would expect then the red streptavidin to bind. So we'd expect to see now red membrane. And in fact, that's what we see. So here's a confocal immunofluorescence image where green is megakaryocyte, blue is the neutrophil nuclei, and the red is streptavidin. And you can see on the neutrophil that's inside the megakaryocyte, it's got red around its membrane, right? It's streptavidin labeled as well. But the megakaryocyte has also acquired streptavidin label. But the megakaryocytes on the bottom here, it's hard to see in this image, but you see that there's no red around them. So the ones that don't do enteropoiesis are not red, and the ones that do enteropoiesis are red. So membrane proteins from this first cell transfer to the second cell. Now, does this go all the way? So remember, megakaryocytes turning their membrane into platelets. So can you make neutrophils? Do neutrophils transfer membranes to platelets? So to do this, we used a trick. The problem is megakaryocytes don't make platelets well in vitro. So you have to really stick them into mice and then watch what happens. So here we labeled megakaryocytes with green in their cytoplasm, and we labeled bone marrow cells like neutrophils with red in their membrane. Then we fed them to each other in paraplesis in vitro, and then if there's membrane transfer and we inject these megakaryocytes into mice, we would expect to see green platelets. That means that they're coming from those donor megakaryocytes with red membrane around them. In fact, so this is just sort of controls here. So you can see we use the CD41 as a marker of platelets. That's our platelet gating. And in fact, that's what we see. So if you look on the left here, the blue are the endogenous platelets. So if you put megakaryocytes in a mouse, it's got its own megakaryocytes, it's making lots of platelets, right? But then you can look at green to see what are the platelets derived from the donor. And on the first panel is if you feed the neutrophils that are not colored. So then you see, of course, the platelets just look like other platelets, they're just green, right? They're in the same otherwise space. They don't express this red color, salvia maroon. But if you fed them with neutrophils that have membrane, those platelets that are made are almost all red. That is, this is a very dominant kind of reaction where in vivo, the neutrophils are transferring their membrane to the platelets. And we call these hybrid platelets, platelets that have megakaryocyte-derived membrane, but also neutrophil-derived membrane. But of course, this is a very artificial system, right? I've taken things in vitro and shoved them into a mouse. That's a very artificial system. So to tell whether this really happens in vivo, what we did was we generated a chimeric mouse. So here you take a mouse, you lethally irradiate it so it has none of its own bone marrow left, and you can graft one-to-one two kinds of bone marrow. One is wild-type bone marrow that doesn't have any fluorescence, and the other is a marrow called MTMG. That it's a marrow that has red membrane, so tomato-colored membrane, that turns green if it's exposed to a Cree. We're not using that yet. So just right now, this is a cell that we use that has a red outer membrane. And we engraft these two into a mouse, and then we looked at its blood. And you can imagine now, if the platelets are all coming from only one lineage or the other lineage, then we'll see either red platelets or not red platelets. They'll be just all mixed. However, if there are some hybrid platelets, we'll see something in the middle. And in fact, that's what we see. Now, we can only really see ones where red is transferred and not red. So we'll see a small fraction of these. But in fact, that's what we see. We see a small fraction of platelets that are hybrids, that have donor neutrophil membrane and megakaryocyte membrane. Oh, thank you. Oh, that's wonderful. Appreciate it. Thank you very much. I even have a pointer. Ooh, that's nice. I have a pointer. Okay, very good. Thank you. Grazie. So then you can see, you can also see this happen when you take the bone marrow out of that mouse, and you can see that there are some megakaryocytes from one donor and some megakaryocytes from the other donor. And sometimes you see a green, sort of a non-labeled megakaryocyte donor with a neutrophil from the recipient, from the red donor in it. And you can see the same kind of bleeding of the membrane into the demarcation system in that context. So you can watch all this happen in vivo as well. And we think that at least 1% to 2% of platelets are hybrid, and we think that's probably true in us as well, although we don't have any evidence in humans. So if the membrane is potentially if membrane production is potentially limiting in the megakaryocyte, maybe by feeding megakaryocytes with neutrophils, they can make more platelets. Is that true? So we took megakaryocytes and colored them either green or red, so we could tell whose platelets were whose. Fed one group with neutrophils and the other not, and fed them both into mice, and we could watch platelet production from the donor strain. And what we observed was that the percentage of platelets that the platelet production by the cells that were fed megakaryocytes was actually more efficient. So platelet production from paraplesis increases the efficiency of platelet production. Now, we don't know for sure that this is because we donated membrane. It could be for other reasons as well, but at least the phenomenon seems pretty clear. So So the initial observation is that we have a kind of a new cell biology, a new kind of cell and cell interaction here, where the neutrophil attaches, and I haven't showed you how it attaches. We worked that out in the paper. The neutrophil enters the cell. It then enters the membrane itself, enters the cytoplasm itself, goes to the demarcation membrane, feeds its membrane to the donor cell, and then leaves. Okay, so are these platelets, are hybrid platelets functionally different? So it's nice to make a few extra platelets, but does this matter? And so what we did was to look at the character of these platelets. And we found in the original paper that they expressed certain markers, surface phosphatidylserine, which is a marker of what are called procoagulant platelets. These are platelets that are especially likely to make clots. And so we wanted to test whether that was true in vivo. And so we again took our complex system where we feed in green megakaryocytes or red megakaryocytes, cultured them either with neutrophils or with control here, neutrophil supernatants, fed them into mouse, and then looked at the platelets that we see coming out. And we looked at markers of procoagulant platelets, and using either arresting platelets or thrombin stimuli or convulsin, which is a GP6 stimulator. That's a surface stimulator of platelets. And we could see that compared to the neutrophils, the first group is endogenous platelets. The second group here is the cells fed megakaryocytes alone. So these are the ones with imperapolysis platelets. These are with just supernatants, and this is with B cells as a control. So we do see this procoagulant platelets, especially in the context of stimulation. So platelets are different. So if that were the case, you would expect that if you got rid of neutrophils, your platelets should change. And so we took advantage of a system called an MRP8 CRE. So a CRE is a fancy system in mouse biology where you can use a kind of a genetic trick to cut out a particular gene in a particular cell lineage, a cell lineage that expresses a related gene. And so we here use MRP8 CRE. So this is a system that cuts out a particular target gene in neutrophils only. And here we used it actually not to cut out a lineage, but to actually introduce something called a diphtheria toxin receptor. So that this allows us to kill one particular lineage specifically. So this allows us, by giving diphtheria toxin to a mouse, we can delete the neutrophils. And we used this system to be able to delete neutrophils in the blood and in the marrow by using, or just in the blood, or just in the marrow, or in neither place. And what we find is that if we delete neutrophils in the marrow, we change the character of the platelets around them. So we do see fewer procoagulant platelets in the megakaryocytes where we've deleted the possibility of doing imperapolysis by deleting neutrophils in the bone marrow. So then we wanted to see, are these platelets really differently coagulant in functional terms? So here we use a system which we built a couple of years ago called a 2-photon microscope system. So the problem with doing immunofluorescence microscopy in a living tissue is that if you do a normal confocal image like this, you just shoot a laser down and look, and everything in this whole column lights up. And then if you're trying to look at it, it's very confused because you see this big thick layer of everything all lighting up green or red or whatever color you're looking at. So 2-photon microscopy shoots in two different signals that sum at a single point to the point that you get an activation threshold. So with the 2-photon microscope, you get a very focused image here. And because you're using low energy light, you can penetrate deep into tissues. And so this allows you to watch cells moving in vitro. That was in vivo. That was the system that when I showed you the lung with the green cells floating into the lung, that was the 2-photon microscope. And we built such a system also in my laboratory. And so we did, in this case, an imperapolysis system where we induced imperapolysis via peritonitis, and we get the same system. So we add LPS to the mouse to add lipopolysaccharides to induce a kind of inflammatory state to increase imperapolysis. And in fact, if we look in the mouse in the bone marrow, looking here at the skull. That's what this is. You can sort of shave off a little bit of the skull and look with the image from the microscope to see what's going on inside the skull. We can watch imperipolesis happen, and in half an hour, you can see imperipolesis again goes up by about twofold, from 11 events per 30 minutes to 28 events per 30 minutes on average. So now we use this in vivo system to test what's going on in thrombosis. So here we've taken a mouse, we've engrafted megakaryocytes from two different colors. So now we have platelets that are derived from imperipolesis, or at least could be, and platelets that are not. And one is green and one is red. And then we then do an imaging system where we expose a vascular bed of the cremaster muscle. And then we can do a laser injury. And what we're looking at is, how do we see platelets accumulate in that clot? Now, remember, most of the platelets that are accumulating in the clot are still the host platelets. So just a few fractional platelets are there. But we're now looking to see, is there a differential between the platelets that are imperipolesis and not? So I think I have to click this manually. Let's take a look. Okay, so here, the blast that you see initially, that is the laser hitting it. And now we're looking for green imperipolesis-derived platelets compared to red non-imperipolesis-derived platelets. And in the platelets that have left the circulation, there's been a hole in the blood vessel induced by this laser, there are about equal numbers of green and red, because that's about the numbers we have. But if you look at the ones that have accumulated in the clot, they're preferentially green. So that tells you that imperipolesis platelets in this in vivo setting, head-to-head, have a comparative advantage in making clots. That is, platelets are these actually thrombogenic in this context. So this tells us the next part of the story. So now we've tested the platelets. We have these hybrid platelets. These platelets have a special function, that they're procoagulant. And this, in a way, makes sense to us. So we know that imperipolesis is induced during inflammation. Inflammations are threat states. You can imagine bleeding is something that happens in a threat state. So you might want to be better at preventing yourself from bleeding in this context, and that is what we think happens. Now, the last part of the story I want to talk about is what happens to the neutrophil. Because remember, the neutrophil is leaving. And so what does it do when it leaves? So the trick is that it actually takes some megakaryocyte bits with it as well. So this is a still from the same video that I showed you before. And I focused you earlier on the green part here of what the neutrophil left in the megakaryocyte. But here's red, the neutrophil has taken with it. So it's taken some megakaryocyte membrane as well. And in fact, if we look at our EM images, we see that happening also. So here's a neutrophil in an imperisome. Here's the neutrophil, here's the megakaryocyte. Here's the imperisome border, and it's actually full of these exosomes generated by the megakaryocytes. Neutrophils don't make exosomes like this, so these are megakaryocyte exosomes. And the neutrophils, you can imagine, they're sort of taking these up as they go through the circulation, as they go through imperipolesis. And here's, EM is always static, right? So you have to sort of paint a picture in your mind of what's going on. But at least the picture we paint ourselves is that these are neutrophils taking up these little dots here. So the neutrophil membrane, for example, is engulfing this bunch of megakaryocyte exosomes. And we can watch this happen in our system, our confocal system. So here we can track, say, this neutrophil as it approaches the megakaryocyte. Notice it doesn't have any green in it at this point. As soon as it enters, it starts to take up green little bits. And as it leaves, it takes those green bits with it. And they essentially all do this. All the megakaryocytes that pass through imperipolesis take up the little membrane. And we can actually see these in vivo as well. So here we've used these MTMG mice, where we've bred them to a mouse that has a Cree that's specific for megakaryocytes and platelets. So now everything's red except for megakaryocytes and platelets that are green. And now we can, so in these systems, we can see in neutrophils that have migrated to the peritoneum, a fraction of them, a small fraction, but a fraction, have these kind of bits in them that look like what we see in vitro. Now, we don't know that those are megakaryocyte bits or platelet bits, we can't tell, but they look to us like what we saw in vitro. And we can see these also in inflamed tissues. So this is a mouse peritoneum introduced with inflammation induced by LPS injected into the belly. And again, we see some of these neutrophils with what look like these megakaryocyte bits inside them, megakaryocyte exosomes. Does this change their behavior? Looks like it does. So if we culture the neutrophils with megakaryocytes and then put them in a transwell system where they have a chance to migrate to our chemoattractant, leukotriene B4. The ones that are cultured with live megakaryocytes, migrate better compared to those that are cultured with fixed megakaryocytes or just megakaryocyte supernames. And we can actually visualize this using microscopy again. So in this system, we have three chambers. So in the middle chamber, we have a collagen matrix in which megakaryocytes are embedded. That's here. So we have this little slice here in which you have collagen matrix with megakaryocytes in them. You put marrow cells in one side and a chemoattractant in the other, and the neutrophils are coursing from one side to the other, and you can watch them. And so what we see in this context is that the neutrophils coming from one side are sometimes passing through megakaryocytes. This is confocal imaging, so we know that it's through and not over and not under. And then we can look to see as they pass through these cells, we can watch the same process happen. They begin to take up these megakaryocyte exosomes. And we can see that the ones that have megakaryocyte exosomes are actually traveling faster than the ones that are not. So the CD41 is the marker of megakaryocyte membrane here. So CD41 negative ones are traveling slower than the CD41 positive ones. Now, of course, this could be you don't know if this is cause and effect. Is this just because they happen to be the faster ones happen to go through megakaryocytes better? So to do that, we can use imaging to track individual cells. And so here you can, I'll show you focus just on the left here, where we can see here each neutrophil is now rendered as a point, and each megakaryocyte is rendered as a space. And you can actually watch these cells go individually into cells. So look at the yellow neutrophil here, it's passing into a megakaryocyte. And you can do this in 2 dimensions. So you can see it, you can make sure that it's really passing through. And now you can watch an individual neutrophil before it goes into a megakaryocyte and after to see what's happened to its speed. And so if you look at ones that have just touched the surface but never entered a megakaryocyte, those cells are moving the same speed. They don't increase their speed at all. So this is before they moved in, this is after they moved in. There's no difference in speed at all. But if you look at the ones that went through the megakaryocyte, they traveled normal speed before and they've accelerated afterwards. And that's a highly significant effect. And we think this is metabolically related, and we have good evidence for that. One piece of evidence there is that if you look at what's in the megakaryocyte microparticles, there are a lot of different components, but they include a lot of metabolic enzymes here. I think I'll skip this, but in any case, we can show that there's a change in metabolism. So again, does this matter in vivo? Here we've taken neutrophils now that have either seen or not seen megakaryocytes, put them in together, compare green versus red, and see which one migrates better. And again, in both LPS peritonitis and IL-1 beta peritonitis, those that have been exposed to megakaryocytes migrate better than those that haven't. So it looks like there's a migratory advantage. And we can see this happen. Now, the trick for us, and one of our big challenges, is that we can't block enteropoiesis specifically in vivo. We don't have yet the pathway to know how do we stop it selectively. So what we've done is instead delete the megakaryocytes for a short period of time. So using this PF4-Cre induced diphtheria toxin receptor system, we can induce a diphtheria toxin receptor expression only on megakaryocytes. Then we give diphtheria toxin receptor, they're gone within 24 hours. Now, of course, platelets also start to go down, and platelets are necessary for neutrophil migration. But for the first couple of days, you can actually see that you have no megakaryocytes, and your platelets are pretty good. There are more than 500,000 per microliter, which we think is probably enough. So then we interpret things that happen when you delete the megakaryocytes in the system to what the effect of enteropoiesis. And so, for example, in this context, we can see that if you delete the megakaryocytes, you don't see any change in the marrow neutrophil numbers. You don't see any change in the peritoneal neutrophil, in the circulating neutrophil numbers, but the peritoneal numbers drop by about half. So you impair neutrophil migration in this context. You can also do that in another model. This is a Pseudomonas model, where we've given the mouse either diphtheria toxin or not. We introduce Pseudomonas into the trachea, sort of a pneumonia model, and then we detect what happens with or without megakaryocytes. And again, we get very similar results. So there's not a change in the marrow neutrophils or in the circulating neutrophils, but then the lung neutrophils are dropped by about half. Now, this is a little surprising that they drop this far. So we would have expected a small change, not a 50% change, because we don't think that all the neutrophils are doing this. So file that one away for just a second. Why is it that the drop is so profound in this context? And we think that has to do with the fact that there's something special about the time when neutrophils are leaving. So if you look at the neutrophils are leaving the bone marrow. So here again is still image from these three-dimensional videos that I showed you before. So here we have this green megakaryocyte pressed up against a white sinusoid. And right between them is this neutrophil right there. So it's sitting in that interface space. So it looks like it might be going this way or it might be going this way. We don't know. So yeah, here's a 3D version of that image. You can see here the neutrophils right at that interface between the blood vessel and the megakaryocyte. And in fact, it looks like it's leaving. So this is a video of a mouse, a bone marrow, where the green is the megakaryocyte, the red is the neutrophil. And the red will be turned into pink when it's inside the space of the megakaryocyte. So, and then you'll see it migrate into the space. You'll see it move around a little bit over the course of a few minutes inside the megakaryocyte, and then it will pop away. And what it's doing is leaving into the sinusoid. So it turns out that this paraplesis, at least in some cases, and probably in most cases, is an egress route for neutrophils from the bone marrow. So in a way, this is the last thing that megakaryocytes, that the neutrophil's seeing in the bone marrow. So it's a chance for the megakaryocyte really to program the neutrophil. So I won't bother with that. Okay, so we think that now comes a different kind of topological question. So imagine you're a neutrophil. You're in the cytoplasm of a megakaryocyte, and you need to get out. How do you do it without ripping a hole in the side of your host cell? And so the answer came from looking, at least we think the answer, came from looking at an early video of our 3-well system. Here we're watching neutrophils pass through. Here it's blown up a little bit. So here's red neutrophils passing through this green megakaryocyte. And if you watch, something sort of special is about to happen that was noted by Pierre Cunin, who was the postdoc at the time when he was looking at these images. What he noted was that. So here you have a neutrophil that's leaving that looks like a comet. It looks like it's got this green tail. And what is that? It turns out that green tail is what we call now a megakaryocyte trail. It's a new structure. So as the neutrophil's leaving, here it's the red neutrophil, it's pulling this long strand of green megakaryocyte membrane with it. And what that does is it allows it, when it finally pops off the end, it's not creating a big neutrophil-sized hole. There's a little pinpoint that has to just close up. The membrane just has to close in order to leave the cytoplasm of the megakaryocyte intact. And it looks like pretty much all the neutrophils leave this way. And if you look at this end of this video, all the neutrophils that have left these megakaryocyte trails. And the megakaryocyte looks like this big spider. So as it has all these trails leaving from it. So these trails are made up, as far as we can tell, exclusively of megakaryocyte membrane, not a neutrophil membrane. Here we use a neutrophil membrane protein and a megakaryocyte membrane protein. It looks like it's only the CD41. These cells do both those little dots inside, those exosomes, and this membrane tail. And the neutrophils seem to like to attach to this membrane. There doesn't seem to be any chemoattraction features, but the neutrophils like to roll on this membrane because they express different markers. And this is not so important, but the point is they express different adhesion molecules that we can stain on these trails. So these look like kind of tracks for other neutrophils to follow. And so to see whether that's true, we took neutrophils that have these trails and put them, this membrane stuck on the end, and added them to endothelial cells, and we can watch them migrate in vitro. So here's a neutrophil with a megakaryocyte bit stuck to the end, plated on a bed of endothelial cells. And you can see it actually attaches that megakaryocyte membrane to an endothelial cell, and then can sort of then starts to wander off. It leaves a little mark. And that mark actually can be quite involved. So here's another version of this. So here, blue is nuclei, but red is megakaryocytes, green is the membrane. You see here a neutrophil leaving this megakaryocyte trail on its tissue. It's leaving this long, thin line. It's going to go around and play with its own membrane here for a second, and then it's going to take off. And now another neutrophil actually can come in and play with that same membrane, and it's going to branch it. You can see it's grabbing the membrane. Second. It's going to pull that membrane. So it can use this to sort of label a space. And so we think that, and if you look at where other neutrophils attach, they attach preferentially to areas where megakaryocyte membrane has been attached. So we think this is a way for neutrophils to mark a place where other neutrophils should go. And if this is true in vivo, and these are very hard to find in vivo because everything's green if you look at an MTMG mouse that has PF4, what we think is happening is that the neutrophils are marking the space and allowing other neutrophils to follow them. So even if there are relatively few neutrophils that have gone through emperipolesis, those serve as pathfinders for other cells. So we think that the outcome here is that neutrophils are being reprogrammed. Migration is improving, marrow egress is giving a new pathway, maybe improving, megakaryocyte trails are happening, and pathfinder neutrophils are happening. So just to summarize, so what's new in what I've just told you? So this is a new kind of cell-and-cell interaction that passes via the cytoplasm. It's a new kind of cell or cell particle, which is the hybrid platelet, which is a platelet that's programmed by the neutrophil. And we think that's probably a very dynamic process that's probably very interesting. We have the idea that maybe angry neutrophils make angry platelets, but we'll see if that's true. There's a new kind of information transfer between immune and hematopoietic systems where the megakaryocyte can talk to the neutrophil and also vice versa. I didn't emphasize that there are two kinds of emperipolesis, but we look at two speeds. You can see there are two different processes going on here at least. There's a new cellular structure we call the MK trail, and we think this is a new neutrophil state, which is what we call the pathfinder neutrophil, which is one that's guiding the way for other cells and improving overall migration into the tissues. So I'll stop there. Pierre, Roxanne, Julia, and Gung Ho are the people primarily responsible for the work, and I'll move back to here and then take any questions. Thanks.